Have you ever been told that all enzymes are proteins? Well, today we're going to look at two incredible discoveries that totally flip that rule on its head: ribozymes and abzymes.
You know, we all learned that biology textbook rule: enzymes are proteins, proteins are enzymes. End of story. But what if that's not the whole picture? What if RNA, the cell's famous messenger molecule, could also be a catalyst? And what if we could actually engineer our own antibodies to do the exact same thing? Let's dive in. So, first up, let's talk about a discovery that literally rewrote the textbooks on how cells work. I'm talking about the catalytic power of RNA.
Okay, so meet the ribozyme. This amazing little molecule is an RNA that completely shatters the old rule. Just like a protein, it folds itself up into a very specific 3D shape, which creates an active site that can speed up chemical reactions. It's not just a passive messenger, it's an active molecular machine.
And listen, this wasn't just some minor finding. The discovery of ribozymes was so revolutionary that its pioneers, Sidney Altman and Thomas Cech, were awarded the Nobel Prize in Chemistry for it back in 1989. It really did change our whole view of RNA's role inside the cell. So, where do you find these incredible molecules? Well, they're all over the place. They're in viruses, they're in our own cells, and they're even at the very core of how proteins get made. Let's look at a few really game-changing examples. For instance, check out the hammerhead ribozyme. You can see from its unique shape why it gets that name. It's found in some plant viruses, and it basically acts like a pair of molecular scissors, snipping itself out of a longer RNA chain. This is absolutely critical for the virus to replicate, and it's a perfect example of how structure defines function, even for an RNA molecule.
But here it is. This is the most mind-blowing example of them all: the ribosome. You know, the massive cellular machine that builds every single protein in your body is fundamentally a giant ribozyme. Yeah! For decades, scientists thought the proteins in the ribosome were doing all the heavy lifting. But now we know it's actually the ribosomal RNA, the rRNA, that forges those peptide bonds. The very heart of making proteins is an RNA-catalyzed reaction. Just think about that for a second.
The fact that RNA can be an enzyme doesn't just change how we see the modern cell, it gives us a huge clue about the very origin of life itself. And this leads us to the super fascinating RNA world hypothesis.
The RNA world hypothesis basically paints a picture of early life where RNA was the star of the show. It did everything. It stored the genetic information like DNA does now, and it catalyzed the chemical reactions of life like proteins do now. The ribozymes we see in our cells today are probably like molecular fossils from that ancient time, a time when RNA was king. All right, so if nature can make RNA into an enzyme, it kind of makes you wonder, right? Can we humans do something similar with other molecules? And that brings us to our second topic: engineering catalytic antibodies, also known as abzymes. So what's an abzyme? Well, it's an antibody that we've cleverly tricked into acting like an enzyme. Think of a chemical reaction like pushing a giant boulder over a hill. An enzyme's job is to make that hill smaller. And it does that by grabbing and stabilizing the boulder right at the very top, what we call the transition state. Abzymes are engineered to do that exact same thing. So how do you even make one? The strategy is just brilliant. First, chemists build a stable molecule that looks exactly like that really unstable transition state. Then, they inject this look-alike molecule into an animal. The animal's immune system, thinking it's a foreign invader, produces antibodies that are a perfect fit for that specific shape. When you isolate those antibodies, they'll now bind to the actual transition state of the real reaction, stabilizing it and making the whole thing go way, way faster.
And I gotta tell you, this is way more than just a cool lab trick. Both of these things, ribozymes and abzymes, have absolutely enormous potential as tools in medicine. I mean, just imagine designing ribozymes to act like those gene scissors we talked about. They could hunt down and destroy a specific messenger RNA, which could silence a faulty gene or even stop a virus in its tracks. And on the other hand, we can engineer abzymes for incredibly precise jobs, like dissolving dangerous blood clots or activating a chemo drug only when it reaches a tumor.
And of course, none of this incredible molecular engineering would even be possible without the raw power of computational biology.
This is where bioinformatics really shines. We can use powerful computers to predict the incredibly complex 3D shapes of RNA just from its sequence. We can model exactly how these molecules work, down to the atomic level. And this isn't just about understanding what's there. It's about designing brand new, custom-made ribozymes and abzymes to fight specific diseases.
So if you take one thing away from all of this, it should be this. The world of biological catalysis is so much richer and more amazing than we ever thought. It is definitely not just for proteins.
So RNA can be an enzyme, and we can actually make antibodies become enzymes too. It's just a powerful reminder that those simple rules we learn in biology class are often just the first chapter in a much more fascinating story. It really makes you wonder, what other biological rules are just waiting to be broken?